I am Masanobu Kano, Professor of Department of Neurophysiology. I will explain the outline of our current research projects. Brain consists of numerous neurons, nerve cells, and glial cells. And neurons are interconnected with the structure called synapse. Therefore, for understanding brain function, it is crucial to elucidate function of synapses, and it is also important to understand how mature neural circuits are formed during development. We use electrophysiological technique to record activity and responses of neurons and glial cells in brain slices by using patch clamp technique. And also we record calcium responses from brain cells in vivo by using two-photon microscope or miniature endoscope. The first research interest is retrograde synaptic modulation by endogenous cannabinoid. Action potentials propagate along axons to reach the presynaptic terminals, and then neurotransmitters such as glutamate or GABA are released and binds to the postsynaptic receptors, and then again converted to electrical signals. When the postsynaptic neuronal activity is elevated, signals are released from postsynaptic neurons to suppress the further transmitter release. In 2001, our group and two other groups in the United States simultaneously reported that endogenous marijuana-like substance called endogenous cannabinoid, N-cannabinoid, are released from postsynaptic neurons to bind to its receptor, CB1 cannabinoid receptor, on the presynaptic terminal to suppress further transmitter release. Afterwards, we identified 2-arachidinoid glycerol, 2-AG, is the endocannabinoid that mediate retrograde signal at synapses. We have elucidated the basic mechanisms of 2-AG-mediated retrograde synaptic modulation. Because the molecules involved in 2-AG signaling are distributed widely in the brain, 2-AG is considered to be a ubiquitous neuromodulator in the brain and contribute to a variety of brain functions and dysfunctions. We are now investigating how 2-AG signaling is involved in these brain functions and dysfunctions by using conditional knockout mice of these 2-AG signaling molecules. The second research interest is synapse elimination in the developing brain. This schematically shows how the functional neural circuits are formed during development. Initially, around birth, neurons form redundant but immature synaptic connections to target cells. During postnatal development, some synapses are strengthened and remain, and others are weakened and eventually eliminated. This process is called synapse elimination or synapse pruning. We use cerebral climbing fiber Purkinje cell synapses as a model system to study the mechanism of synapse elimination. We found that single climbing fiber input is selectively strengthened during first postnatal week, which is called functional differentiation, and then the strengthened climbing fiber, so-called Wiener climbing fiber, can extend its innervation territory along dendrite, which is called dendritic translocation, and in parallel, The somatic synapses are eliminated in two distinct processes called early phase and late phase of climbing fiber elimination. By the end of the third postnatal weeks, most Purkinje cells become innervated by single climbing fiber on the addendra. We have been searching for molecules involved in climbing fiber synapse elimination. This slide summarizes how multiple molecules are involved in elimination of the so-called loser climbing fiber and strengthening of a single winner climbing fiber. If you are interested in our lab, please contact us from this URL. Thank you very much for your attention.